Hi everyone, my name is Michael Seeler, and today I'll be presenting some results of my study modeling the gut microbiome's resistance and resilience to climate change and infection in zebrafish. Anthropogenic climate change is causing increasing global temperatures and impacting wildlife and human health. Given the gut microbiome's central role in supporting host health and homeostasis by mediating the effects of exogenous stressors, such as those exacerbated by climate change, there's an urgent need for understanding how climate change will impact the gut microbiome to influence host health. For instance, ecologists anticipate the range of infectious agents, such as intestinal parasites, to expand as global temperatures increase. Of particular concern are the increased intestinal parasite burdens on aquatic organisms experiencing increasing water temperatures. Given the gut microbiome interfaces with exogenous stressors, such as those as intestinal parasites, there's a need to clarify how climate change may modulate the gut microbiome infection axis. We hypothesize that the gut microbiome's response to an exogenous stressor will vary in terms of its resistance or sensitivity depending on the host's environmental conditions. Specifically, we anticipated that the gut microbiome would be most sensitive to an exogenous stressor when the environmental conditions vary substantially from their baseline conditions. To evaluate the gut microbiome's response to an exogenous stressor across environmental conditions, we leveraged the zebrafish model system due to the organism's unique advantages to interrogate the gut microbiome. Zebrafish are an increasingly popular model organism to study vertebrate-associated microbial communities of the gut microbiome due to their well-established high-throughput methods for microbiome manipulation and analysis. Additionally, zebrafish can tolerate a wide spectrum of temperature ranges from 4 to 40 degrees Celsius, despite their thermal optimum being 28 degrees. So this affords us the ability to evaluate their microbiome's response across a gradient of environmental conditions. And prior work in zebrafish has independently investigated the impacts of altered temperatures and parasite exposure on their gut microbiomes. But the interaction of these factors and their temporal impacts have not been studied. To clarify the potential impacts of climate change on the gut microbiome and host health, we sought to determine how the zebrafish gut microbiome varies across environmental conditions of increasing water temperatures, as well as measure the temporal infection outcomes and gut microbiome response across increasing water temperatures when exposed to the intestinal parasite Pseudocapillaria tomentosa. To do this, we reared zebrafish to adulthood and then acclimated them to one of three water temperatures, 28, 32, or 35 degrees Celsius. After collecting baseline fecal microbiome samples and assessing intestinal histology, we exposed a cohort of fish from each temperature group to the parasitic worms. Between 14 and 42 days post-exposure, we collected subsequent fecal microbiome samples and measured worm burden via histological and wet mount assessment. First, we sought to determine how water temperature impacted infection outcomes in parasite-exposed fish. Here we have a figure that will show the total number of worms detected per fish by temperature group across time. On the y-axis, we have number of worms detected per fish, and on the x-axis, we have time written as days post-exposure. Number of worms per fish will be displayed as diamonds on the plot. Currently, I'm only showing the results of worm counts between the temperature groups at our baseline sampling time point where all fish have not yet been exposed to parasites. Next, at 14 days post-exposure, we see that fish in the 28 and 32 degree groups have detectable worms. And by the conclusion of our experiment, we see that infection trends in an increasing manner in the lower water temperature fish, while 35 degree fish remain virtually uninfected except for one instance at 28 days post-exposure. Together, these results suggest that higher water temperatures may be protective against infection, but more work is needed to fully understand the parasitic worm's ability to reproduce and infect fish at each of these water temperatures. Given the differences in infection outcomes between the fish reared at different water temperatures, we next sought to determine how the microbiome varied across the water temperatures when exposed to a parasite. To do that, we measured the microbiome's gut microbial diversity, community composition, and then investigated the association between infection outcomes and these microbiome measurements. First, I analyzed the gut microbiome variation in terms of gut microbial alpha diversity. And here I'm showing a figure displaying the median alpha diversity scores as measured by the Simpson's index for each temperature and treatment group across time, with violin plots indicating the amount of variation for each group. On the y-axis, we have alpha diversity scores scaled from 0 to 1 based on minimum and maximum observed values. And on the x-axis, we have time written as days post-exposure. Median alpha diversity scores will be displayed as circles or diamonds on the plot corresponding to control or exposed treatment groups with corresponding colors indicating water temperature. And here I have the results for our baseline sampling time point at 0 days post-exposure. And as expected, we see no statistically significant differences between the treatment groups prior to parasite exposure. By day 14, we start to see temperature-dependent trends in diversification of the parasite-exposed fish reared at 28 degrees Celsius water temperature. Notably, the 28-degree parasite-exposed fish display increased variation seen in the wide spread of the violin plot. From 21 to 28 days post-exposure, the alpha diversity scores of these 28-degree parasite-exposed fish are significantly less than the control fish. These results indicate sensitivity of the gut microbiome to parasite exposure at 28-degree water temperature. Whereas the trend in alpha diversity scores for the 32 and 35-degree parasite-exposed fish are relatively temporally stable and do not differ to their controls. These results indicate that the gut microbiome's resistance to parasite exposure at higher water temperatures. By the end of the experiment, we do not find statistically significant differences between treatment groups across each of the temperature groups, but there is substantial variation between or within the parasite-exposed groups of the 28 and 32-degree fish, indicating sensitivity, and this is likely contributing to our inability to detect statistically significant differences. Collectively, these results suggest that in terms of alpha diversity, the gut microbiome's sensitivity to parasite exposure is a function of water temperature. Next, we sought to determine how parasite exposure affected the gut microbiome community composition across increasing water temperatures and time. Here, I'm showing beta diversity ordination plots faceted by water temperature. The X and Y axes labeled MDS1 and 2 represent the percent of variation in Bray-Curtis dissimilarity distance derived from multidimensional scaling. Each point on the plot represents a unique sample, and the points are colored by water temperature. Again, circles represent control samples, and diamonds represent parasite-exposed samples. And for those of you not familiar with these plots, the closer the points are to each other indicates that microbial communities of those samples are more similar to one another, and if the points are further away, that indicates that the communities are more different. This figure is only showing the results for a baseline sampling time point prior to parasite exposure. And Permanova tests did not find statistical differences between the centroids of the treatment groups within 28 and 32-degree water temperature groups prior to parasite exposure. But it did detect a significant difference in the 35-degree fish, indicating that gut microbial communities of fish reared at 35 degrees water temperature differ from one another. And together, these results indicate that gut microbial community compositions at lower water temperatures are initially more similar to one another prior to parasite exposure. And finally, skipping ahead to 42 days post-exposure, we see that fish gut microbial communities become statistically different between the treatment groups across temperature group by the conclusion of the experiment. Additionally, we detected elevated beta dispersion levels in the 28 and 32-degree parasite-exposed fish. These results indicate that these parasite-exposed fish reared at the same temperature are less consistent to one another. And collectively, these results indicate that gut microbial communities of fish reared at lower temperatures are more sensitive and inconsistent to the effects of parasite exposure relative to fish reared at higher water temperatures. Because I observed variation in the gut microbiome response to parasite exposure across water temperature and time, I then wanted to see how this variation might associate with the infection outcomes I discussed earlier. To do that, I first plotted infection counts by alpha diversity score for parasite-exposed fish. Infection counts are on the y-axis, and alpha diversity scores are now on the x-axis. Each diamond represents a unique sample. I found an interesting nonlinear trend between high infection count and alpha diversity. Infection is indicated by the darker filled diamonds, and the samples with no infection are transparent. And we see that fish with the highest infection burden tend to associate with either the highest or lowest alpha diversity scores relative to uninfected fish. I then wanted to see if these infection trends manifested in beta diversity space as well. I observed samples with the highest infection burden were furthest away from the uninfected samples, suggesting that microbiome community composition of highly infected fish differed from uninfected fish. I then grouped the samples with infections by highest or lowest alpha diversity scores. This grouping manifested into two distinct clusters in beta diversity space as well. I found that high alpha diversity group was mostly composed of 32-degree fish, while the low alpha diversity group was predominantly composed of the 28-degree fish. These results highlight an important environmental contextual relationship between infection outcomes and microbiome response. In particular, these results have implications for how we think about ecological concepts as the Anna Karenina principle in the context of climate change's impacts to the gut microbiome. According to the Anna Karenina principle, dysbiotic or unhealthy microbiome communities display characteristics of stochasticity and a high degree of variation in the microbiome measurements, suggesting an unstable state, similar to what we see here, where in both plots, samples with high infection are seemingly randomly scattered and not following a deterministic pattern or linear trend. However, when we consider the environmental conditions of a gut microbiome's response to a stressor, the Anna Karenina principle seemingly diminishes and multiple alternative stable states emerge, thus demonstrating that when we carefully consider the environmental context of a gut microbiome and its host, we can reveal otherwise latent or obscured patterns in microbiome responses to an exogenous stressor. And elucidating these patterns is critical to uncover potential underlying causal mechanisms and for evaluating and predicting microbiome stability and host homeostasis to exogenous stressors. In conclusion, we found that highest infection burden and sensitivity to parasite exposure was observed in lower water temperature reared fish, while gut microbiomes of fish reared at higher water temperatures were more resistant to the effects of parasite exposure. These findings support the notion of the gut microbiome's buffering capacity against exogenous stressors, and that this buffering capacity may be environmentally contingent. However, it may also be the case that increasing water temperatures impacted parasitic burden or parasite worm infectivity, and consequently the microbiome was unaffected. Additionally, our study did not consider the contributions of the host and their immune system, which may interact with the microbiome to resist infection. So ongoing work is investigating the parasitic worm's ability to reproduce and infect fish across a range of water temperatures to clarify the causal direction of the microbiome infection axis. I'm also developing novel methods to further tease out nuanced microbiome measures of temporal stability. Overall, our study demonstrates that anthropogenic climate change may have unanticipated and environmentally specific impacts to ecosystems and organismal health, which has important implications for how we as researchers and society develop context-appropriate approaches to study, address, and resolve the impacts of climate change to ecosystems and individual health. And with that, I'd like to thank my collaborators and funders for supporting my research efforts. And I'd like to thank you all for your time and attention. Thank you for watching. And if you have any questions, please leave a comment or shoot me a message. I will happily respond to you as soon as possible. Thanks.